Welcome to biology. This presentation introduces you to the next unit, which deals with cell cycle and DNA replication. So we'll be going into some more depth with DNA, which is a nucleic acid, one of the four types of categories of biomolecules that we learned about in Unit 2. And we will also be looking at the cell cycle. So as far as characteristics of living things are concerned, we're looking at growth and development and reproduction. We're going to be discussing some of the reasons that cells would go through cell division, but one of the main reasons that a cell goes through cell division is if it gets too large. If a cell gets too big, then it will trigger itself to divide. And one of the reasons that it does this is directly due to this thing called surface area to volume ratio. And so the larger a cell becomes, the more volume it has, which means it has less surface area. So you're going to learn how to calculate surface area and surface area to volume ratios. But for now, what I want you to know is that the higher surface area a cell has, the more efficient it is. So if we're talking about processes that you guys just learned about, such as cellular transport that occurs on the plasma membrane, if the plasma membrane has a high surface area to volume ratio, then it makes it very efficient at moving things in and out of the cell to regulate its internal and external environment. So that's one of the reasons why cells divide, is because they become too large. So this looks at the cell cycle in its entirety, and this is not something that you're going to know everything about it day one. We're going to be spending some time with this, but if you look at this, we've kind of got this divided into two main components, and we can draw this out later on. But one is the period of growth, and the other is a period of division. And so we're going to take a look at the period where the cell is growing and preparing for division first. And that is kind of outlined if I drew a line kind of dividing the halves here, then on this side would be what we call the period of growth, and on this side would be what we call the period of division. So the period of growth is known as interphase. So this is what happens prior to the cell actually going through division. Interphase is divided up into 3 steps. So think of this whole cycle as moving in a clockwise direction. And so we're starting here first in G1. And so when you see that G, I want you to use the G as an alphabet analogy or an association. And I want you to think of that G as growth. So we just mentioned that one of the things that triggers cell division is that the cell grows. So the cell is getting larger. It has taken in organic molecules. It has taken in water. It is increasing that internal volume. And so it is growing and getting ready to prepare for cell division. In step 2 of interphase, and again, we're only looking right now at interphase. That is our only focus. We have the S phase. And that S, when you see that, I want you to think of that S as, I want you to think of this word called synthesis. And you guys have seen that word before. And what that word means is to make. And so what we're going to do is we are going to synthesize the DNA, which is also known as DNA replication. So we're right back to that trend in biology where we have different names that mean the same thing. These can be used interchangeably, DNA synthesis or DNA replication. And what that means is we're going to make a copy of the DNA. At this point in time, the DNA is contained in these structures known as chromosomes, and we're going to dissect this today and talk about and learn about chromosome structure. But basically, in step 2, in that S phase, we have growth that continues, and we have the duplication or replication or synthesis of DNA. The third thing that happens in interphase is the G2, and the cell grows some more and gets ready to divide. So right here, this line right here, division does not start until we get to that point in the cell cycle. So just to recap, we're looking at the cell cycle, and by definition, cell cycle, you start where you end, et cetera, and so on. And it has 3 parts, which is G1, S, and G2. So those are the 3 parts of interphase, which has to occur first. So chromosomes, what are they? You guys have probably heard of chromosomes before, but you may not be familiar with their structure or what they actually are. So chromosomes shown here, and you may have seen this structure represented as just an X. So we sometimes call these X chromosomes or replicated chromosomes or metaphase chromosomes. But basically, they're structures that are made up of DNA and protein. So right back to those nucleic acids that we learned about in Unit 2, we're seeing how 2 of them, DNA and protein, come together to make these cell structures known as chromosomes. And we'll talk about how bacterial chromosomes are different than eukaryotic chromosomes a little bit later. But if you see that X chromosome structure, we're dealing with eukaryotic chromosomes. The word for chromosome comes from some prefixes and suffixes where the chroma means colored and the soma means body. So these are colored bodies. So when you start to look at some cell diagrams and you see our cell here, and then you're going to see a nucleus, and then inside that nucleus, you're going to be able to see, and you won't be able to differentiate these chromosomes, but that is what the chromosomes are, is that dark material that are inside the nucleus, which is why we call them, chromosomes. They're colored. You can see them. If you guys were in class, we would be looking at these through a microscope. And one of the things that you can distinguish looking at a eukaryotic cell through a microscope is the nucleic material. And so these chromosomes have this structure, but we have to keep in mind that if we really zoomed in, if we took a piece of that and zoomed in on it, we get our characteristic double helix DNA molecule. And often we leave out the protein, but it's good to know that it is there, even though when we really focus on a chromosome, we're primarily interested in what the DNA is doing. One of the things you have to know is the parts of chromosomes. And so the whole thing is called a chromosome. This whole structure right here is referred to as a chromosome. Now, each chromosome is comprised of these things called chromatids, and then sometimes we call them sisters. So I'm gonna draw it out so that you guys can see it from start to finish. So with a chromosome, we have our whole structure here, and then so this whole structure is the chromosome, and then we label these guys right here as chromatids, and we put another word in front of that sometimes, and we call them sister chromatids. And the reason why we call them sisters is because they are identical. The reason why they are identical is because we started out with one chromosome, and then we made 2 chromosomes during the process of DNA replication. So since we took that 1 chromosome and made an X chromosome, those chromosomes that we call sisters that are together in this X-shaped structure are identical, and that's why we call them sisters. So we use female terminology to describe things that are identical. So you'll see things referred to like sister chromatids, or you'll see other things referred to like daughter cells. And so when we use those terms like sisters and daughters, we're describing things that are identical. The other thing that I will draw your attention to in this diagram is this structure right here known as a centromere. And notice that kind of looks like center. So think of that as something that's in the center of the chromosome that holds the sister chromatids together. That's the function that it performs. And another thing worth mentioning is that it is also a protein. So even though we are finished with organic molecules, we have learned about carbs and lipids and proteins and nucleic acids, we can't just box that information up and put it on a shelf. We refer back to it with a lot of frequency as we move through these other units. So just keep in mind these chromosomes are made of proteins as well as DNA, even though the DNA is what we're primarily interested in. So, what is DNA replication? Going back to interphase, that period of growth. During that S phase, so remember we had G1 first, and then we had S, and then we'll have G2 afterwards, but DNA replication only happens during the S phase. And what's occurring is the DNA is making a copy of itself. So that's all replication is the DNA making a copy of itself. What you can see over here, and I'll front load you a little bit here. What you can see are an A is with a T, and a C is with a G, and a G is with a C, and an A is with a T. So the point being is that A and T go together, and G and C go together, and you're going to learn these base pairing rules. These are actually called complementary base pairs because they are complements of one another. A and T always pair together. G and C always pairs together. And when the cell divides, since we have this replication mechanism in this base pairing, each new cell gets an identical copy of the DNA, which is extremely important. DNA replication steps. This is how this process occurs. Step 1, the DNA will unzip itself. So we're talking about the DNA that makes up that chromosomes. It'll start to unzip. So think of it like a zipper on a jacket. It starts to unzip, which allows it to open up. And when it unzips, then the 2 chains separate here in step 2. So in step 1, the DNA unzips. In step 2, we have separation of the 2 chains. So keep in mind that DNA is a double helix. Another thing that I'll point out here is all of these things we're going to be learning about, whether it's DNA replication or whether it's, later on protein synthesis, all of these things are biochemical reactions that have to have enzymes in order to work. So DNA helicase, ACE, meaning it's an enzyme, is one of the enzymes that are involved in this process of DNA replication. So this stuff just doesn't occur on its own. It has to be catalyzed by specific enzymes that allow the DNA to unzip itself and these 2 chains to separate. And then there are other enzymes that allow new strands to form as well. Continued DNA replication steps. So it unzips, the strands separate, And then we bring in new nucleotides. So go way back to organic molecules, may seem like a while ago, but we have those structures known as nucleotides. If you remember, nucleotides were monomers of nucleic acids. So, with all the categories of organic molecules, carbs, lipids, proteins, nucleic acids, we learned their monomers and their polymers. So, this is a nucleic acid monomer. Notice that nucleic acid, nucleotides, DNA is found in the nucleus of eukaryotic cells. So, all those words go together, and it's important to remember the structure of those nucleotides. So you've got a phosphate group attached to a sugar that's attached to a nitrogen base. And so when we were looking a second ago at that diagram of DNA replicating, we saw the A's paired with T's and the G's paired with C's. So those letters represent the nitrogenous base of the nucleotide. And remember, nucleotides have those three parts: a phosphate group, a sugar, and a nitrogenous base. So a lot of times we'll use only the letter that represents the nitrogenous base to represent the whole nucleotide, but it's important to remember that the nucleotide actually has two other components in addition to that nitrogenous base. So new nucleotides are brought to make the new strand. And again, we don't have to memorize the names of all these enzymes and understand exactly what each one does, but DNA polymerase. So DNA is a polymer. So polymerase is the enzyme that is going to add these new nucleotides to the original strands. And that's where you see that complementary base pairing that we were describing earlier, where A pairs with T and G pairs with C. Once all of the base pairs have been made, then step 4 right here, the two identical strands will twist back where you started with one original DNA molecule here. You end up with two new double helices that are the exact same copy of the original. If we were looking at this in chromosome form, then what this would look like is this DNA would look like this right here before replication. And then after replication, we would get that characteristic X-shaped chromosome. Okay, so it's important to understand what's happening at the molecular level, shown here, and then also what's happening at the structural level that I just drew out in the form of chromosomes up at the top. The other thing that you need to know about DNA replication is the model that is the correct replication model. And the model that is the correct replication model is what we call the semi-conservative model. Semi, think of that as half. Conserve, think of that as, we save it. So in the semi-conservative model of DNA replication, every time a chromosome replicates itself, then each new chromosome has one original strand of DNA, or what we might refer to as the old strand or original strand, and then it has one new strand. So the DNA molecule that's being synthesized has one old and one new. If you look at the diagram, down here at the bottom, our original or old strands are representing the new strands, or excuse me, the original strands, and then the red is representing the new strands. And so each new synthesized chromosome will have one of the original strands and one of the new strands. So if we were going to draw out the finished product here, what we would get in the end is we would get two strands of DNA, or what we could say two chromosomes or two sister chromatids that would each consist of one original strand and one new strand.